Hello students, we will start our next chapter, that is biotechnology. So when we refer to biotechnology, what does it mean? It means the technique of using, bio means living organisms. So it means the technique of using living organisms to producing some products which may be useful to human beings, useful for industries and all. So we are using living organism in terms of technique. We are using some technology in which we are using the living organisms. So many processes like in IVF, you might have heard about IVF, then developing DNA vaccines, all those come under biotechnology. So when we say about the definition of biotechnology, EFB, that is the European Federation of Biotechnology, European Federation of Biotechnology, they say the integration of natural science. What is biotechnology? They say the integration of natural science and organisms. Organisms, we can say they are cells, their molecules and all. So we are integrating the natural sciences and organisms to produce some products. So that is the definition that is given by EFB, European Federation of Biotechnology, and they say that biotechnology is the integration of natural science and organisms to produce some products or services. So when we talk about the principles of biotechnology, we refer to genetic engineering. Genetic engineering, what does it mean? It is the technique to alter the chemistry of DNA or RNA. You know, DNA and RNA, they are the genetic material. So in genetic engineering, it is the technique to alter the chemistry of DNA and RNA to we are introducing that, altering the DNA and RNA, we are introducing that into a host organism and we are changing the phenotype of the host organism. Just take an example of firefly. What is the main feature of firefly? That it can emit light. Now suppose we want this feature of firefly, that is it can emit light at night. This feature of firefly, we want to introduce that into a frog, suppose. So this frog, we want this frog also to emit light. So what we will do, we will take the gene which is responsible for giving this feature to firefly of emitting light, take that gene and introduce into frog. And so we are changing the phenotype of the frog. So frog is the host organism. So this is what is genetic engineering all about. Then chemical engineering, when we consider, there are some restrictions to chemical engineering, that chemical engineering can only be done in a sterile ambience. So the atmosphere should be perfect, so that the microbes and all, they can be handled properly in a suitable environment. So chemical engineering involves the maintenance of sterile ambience. So when we see the conceptual development of genetic engineering, first we have to understand this, that sexual reproduction is better than asexual reproduction. Why? Because in sexual reproduction, variations are there. We get unique combination, we get different combination, and variations are possible in sexual reproduction, whereas in asexual reproduction we get exact copies. So there is no variation there in case of sexual reproduction. Then second thing that we should remember is genetic engineering is better than traditional hybridization. Genetic engineering is better than traditional hybridization. Why? Because in traditional hybridization, when we are doing traditional hybridization, whatever is undesired also, suppose we want a desired character and we are using that organism, doing some hybridization to produce some desired character. But along with the desired character, some undesirable characters also come in case of traditional hybridization. But in case of genetic engineering, that is not the case. The genetic engineering, we are only taking the desirable gene. Desirable gene, I gave you the example here of firefly. It's a desirable gene that we want is the gene to emit light. So we are only taking that gene of interest. It's called the gene of interest. So that is the desirable gene which we want to introduce into frog. Frog is the host organism, or you can say that is a target organism.